Now, there is some general nomenclature depending on what you are looking at as far as the base versus what we call a nucleoside versus a nucleotide, and if they are in DNA or RNA, and that can be found in this table 8.1. So generally, when we say adenine, guanine, cytosine, thymine, or uracil, we're just referring to the actual nitrogenous base itself. This is this ene ending. You can see if we talk about the nucleoside, which does not have phosphate, we use the sine ending, adenosine. And if we are talking about the nucleotide itself, that's with phosphate, we use adenylate. And this can also be, again, the whole thing can be named based on how many phosphates there are. So for example, if we did ATP, that would be this adenosine triphosphate. So here are the actual structures of the full nucleotide, depending on if you have an adenine, a guanine, a thymine, or a cytosine. And how you name them and their symbols that we can use to talk about their names. So again, if we have the actual nucleotide, we're going to use the adenylate, the 8 ending, that an 8 ending. And here we're distinguishing also what type of sugar it is attached to. So for a nucleotide that has one phosphate on it and uses a deoxyribose, this would be called deoxyadenosine 5' monophosphate. So deoxy is for the sugar, adenosine is the base, 5' is where our phosphate is, and we said that there is one phosphate, that's monophosphate. This could also be distinguished as DAMP, and the letters go with everything we just talked about. D, we always use D if we're using a deoxyribose sugar. So D is for deoxy, A is for adenosine, M is mono, and P is phosphate, so monophosphate here. Now, if it did not have the phosphate attached to it, remember, we would not add that 5' monophosphate naming group on the end of it. And the nucleoside name, the name with no phosphate, would just be deoxyadenosine. So this naming system holds true depending, of course, whether if you have the G, T, or C type of nucleotide attached. Now notice all of these given, A, G, T, and C as examples, are for the deoxyribonucleotides using deoxyribose. I did not include uracil here, and the reason is because uracil will not ever be attached to a deoxyribose because uracil is not found in DNA. So only A, G, T, and C attach themselves to deoxyribose. If we think about ribose, full ribose, now we can consider uridine and not thymine because uridine, U, is found only in RNA, and thymine is only found in DNA. These are named much the same as we just went over, except here we do not use the deoxy part. So for example, for the adenylate nucleotide, this would just be adenosine 5' monophosphate. So no D in the front. And its shortened form would again not have a D. This means we're using the ribose sugar. It would just be AMP. So know your structures, know what the structure means, what it looks like, know the name, this adenosine 5 monophosphate, for example, and know the symbol, know what it means if I say DTMP versus CMP.